It is my great pleasure to introduce today's first speaker and my good friend, Dr. Julia Rogers. Julia and I did our postdocs together, and she is both an outstanding scientist, and it sounds a bit cheesy, but it's true, she's an even better person. Um, and a fun fact, we actually share the same birthday and have had many shared celebrations over the years. And for those who celebrate, today is our two-third birthday. So a fitting day for me to host Julia for a seminar. So some background on Julia. Julia completed her undergraduate degree in molecular biophysics and biochemistry at Yale before moving to Harvard for her PhD in biophysics under the supervision of Martha Bullock. There, Julia did some really fantastic work with a series of incisive and rigorous papers studying complexity in protein DNA sequence recognition, and she was awarded an NSF Graduate Research Fellowship. For her postdoc, she joined the labs of Steve Blacklow and Karen Edelman in the Biological Chemistry and Molecular Pharmacology Department at Harvard Medical School, where she has been studying the NOT transcriptional response with a recent paper just out in Gene Development. And over the way, she has earned numerous awards and accolades, and I won't list them all, but I'll note that these include a fellowship from the Leukemia and Lymphoma Society and the Mosaic K99 Independence Award. Throughout her career, she has also worked to give back and improve the culture around her with many efforts and initiatives, including her postdoc. She served as the co-chair of the department training committee and the departmental task force on diversity, equity, and inclusion. And I saw firsthand the positive impact she had in the department, how well she works with people, both faculty, department staff, and other trainees. She's also just a great person to go for advice, be it scientific or other. And all of this has contributed to her being incredibly well respected and liked member of the department, which brings me to my last point. Julia is currently on the job market, and I think that any department would be lucky to hire her. Outstanding scientist, amazing mentor, and superb colleague. She's going to land somewhere great, and for those of you on search committees, you should do everything you can to have that place be in your department. But I think I've gone on too long, so I'll finish by saying that I'm very excited for Julia's talk today, so please join me in giving a warm, albeit virtual, welcome to Dr. Julia Rogers. Julia, the floor is yours.
Thanks, Ben, for the really nice intro. You're going to make me cry a little bit there. But thanks so much for inviting me to participate in this seminar series. Yeah, I'm really excited to share with you all today the work that I've done as a postdoc, really trying to understand better how Notch signaling induces gene expression. So as you can sort of tell from Ben's intro, I've been really interested in transcription factors throughout my career, and this work is part of my big picture attempts to really understand at a molecular level how transcription factors work. So as many of you may know, transcription factors are the proteins that regulate gene expression, and they can promote transcription at multiple steps during the transcription cycle. So first I'll outline what these different steps are. So we know that before a gene is induced, transcription factor binding sites can often be occluded by nucleosomes, and the transcription start site itself can also be occluded. So one step that a transcription factor can act at is by promoting accessibility around transcription start sites and regulatory elements. So that's shown here by this blue transcription factor in this cartoon, which in this case is recruiting the switch SNF remodeling complex, and that'll come up later. So in addition to this initial step of promoting accessibility, we know that after this, Pol II is recruited to the transcription start site by a lot of the accessory transcription factors. And what's been more appreciated recently is that another highly regulated step is promoter proximal Pol II pausing. So after Pol II is recruited to the transcription start site, it transcribes a little bit downstream and then sits and waits for additional signals, which tells it to go into the to full elongation in the creation of the full gene product. So these signals can be brought in by additional transcription factors like this green guy shown here, which can promote the recruitment of PTEF-B and then release of pause Pol II and production of the full gene product. So in my postdoc, I was really interested in trying to understand what transcription factors, at a molecular level, what transcription factors do, and do different transcription factors promote different stages of this transcription cycle. And the system I chose to work in is the Notch signaling system, and I'll go through a few slides explaining why that was a great system to work in to address this big picture question. So first of all, Notch signaling is really important in biology. It is essential for development of a variety of organs in the human body. I've outlined a few here. And something that I think is really interesting is that in all of these different organ systems, Notch signaling actually induces different gene expression programs in these different cell types. So you'll get different, there are some core genes that are always induced in response to Notch signaling, but you can get different gene expression programs in these different systems. Notch is not only essential during development, but is also important for homeostasis. So in an adult, Notch signaling is also active in promoting homeostasis of a variety of your organs. Because of its importance in these different systems, Notch signaling dysregulation is associated with a variety of types of cancers. And interestingly, given its different roles in different cell types, Notch signaling can either be oncogenic or tumor suppressive, depending on the cellular context. So for example, about 50% of T-ALL or T-cell leukemia cases have activating Notch1 mutations. Or on the flip side, a large percentage of cutaneous squamous cell carcinoma cases have inactivating Notch mutations. So next I'll move on to some of the molecular players in the Notch signaling system so you can understand who I'm talking about and when I get more into the details. So Notch signaling is a juxtacrine signaling pathway, which means it allows cells that are in direct physical contact with each other to communicate. So you'll have a sender cell, which expresses one of the Notch ligands, either in the delta or jagged family, and a receiver cell, which expresses Notch itself. When these two proteins come into contact, that triggers a series of changes near the cell membrane, which allows two regulated proteolysis steps, first by a member of the ADAM family of proteases, and then by the gamma secretase complex, which actually cleaves right on the inside of the cellular membrane, which then allows the intracellular domains of Notch to go into the nucleus, where it forms what's known as the Notch transcriptional complex with two other proteins. One is RBPJ, which is responsible for binding directly to DNA, and the other is MAML, or MAML1 in this case, which is a co-activator and is absolutely required for the expression of target genes. One additional feature that makes Notch a really great system to work in for understanding how transcription factors function is that we can actually toggle the accessibility of Notch to go in and out of the nucleus because there are inhibitors of this gamma secretase complex known as GSI for gamma secretase inhibitors. And using these, I can easily toggle Notch in and out of the nucleus and really see over time how activation of Notch triggers gene expression. So the overall question that I've been really trying to address is once Notch is activated and this transcription complex forms, how does this actually induce the expression of target genes? So a lot of previous work in the field has really focused on the possibility that Notch acts really early in the transcription cycle, like the blue transcription factor that I highlighted in the cartoons earlier. Specifically that it might interact with a switch-NIF complex to promote chromatin accessibility. So the switch-NIF complex is a major chromatin remodeling complex, and it generally promotes accessibility at regulatory regions, both transcription start sites as well as enhancers. We know that there are a lot of genetic associations between different components of the switch-NIF complex and Notch signaling activity. And in a lot of like pull-down assays and such, we see a lot of components of the switch-NIF complex coming down with Notch. So it seems like there might be some interaction there. However, we also know that switch-NIF can actually be bound at Notch-responsive promoters before Notch signaling is ever really activated. And then also more conceptually, the fact that Notch is leading to the induction of different gene expression programs in different cellular contexts sort of suggests that Notch might not have such a directive role towards chromatin accessibility, but might be more responding to the chromatin landscape that it sees. So I was really interested in trying to address if Notch really acts like this, or more broadly, what does Notch actually do to induce transcription? A lot of the previous work trying to address this sort of question really compared steady state conditions where Notch had either been on for a long time or off for a long time. But to really understand what Notch signaling does to transcription, I needed a dynamic system and dynamic approaches. So first I'll talk through the cellular system that I worked in. I worked with our collaborators in John Astor's lab at the Brigham, who had recently developed a squamous cell carcinoma model cell system that was a great system for me to work in to understand how Notch activates transcription. So as I mentioned earlier, in squamous cell carcinoma, Notch is often inactivated. So our collaborators started with a cell line with inactivating Notch mutations and put in back a truncated version of Notch1. So this version of Notch is missing all of its extracellular domains. So it's actually ligand independent, so you don't need to add a sender cell or anything, but it's still gamma secretase inhibitor sensitive. So when we culture these cells in the presence of GSI, which we do all the time ever since we added in this transgene, Notch remains off and tethered at the membrane because it can't be cleaved to be released into the nucleus. When we do a GSI washout, so change the media and remove this inhibitor, Notch is then able to go into the nucleus, and the cells over a course of days begin to differentiate into a keratinocyte-like fate. In the course of just a few hours, though, we actually see the induction of a lot of Notch target genes. So this means that I can look within really short time points and start to see how Notch induces transcription. When we do what's known as a mock washout, so change the media but don't remove the inhibitor, the cells basically act like nothing had happened. So using this system, I first needed to identify exactly which were the Notch target genes in this system, so that I knew where to look in the genome to see what happens to chromatin and transcription. So to do that, I used an approach called TT-seq, or transient transcriptome sequencing, which is a metabolic labeling approach that tells us which are the newly transcribed RNAs over short periods of time. So I did this approach looking at 2 different time points after inducing Notch signaling. And importantly, I also did a mock washout control to make sure that the genes that I see that are induced are specifically induced in response to Notch. So using this approach, I identified a high confidence list of 61 genes that we know are induced by Notch in this cell type. You can see that some of them start to come on within just an hour, and many of them are maximally induced at 4 hours. Within this group of genes, we see a lot of genes that are expressed, commonly expressed in response to Notch signaling, such as these Hess genes, as well as some cell type specific targets. And overall, when we look at the Go terms that categorize these genes, we see there's a lot of things involving development or differentiation, which is consistent with what we know Notch does in a variety of systems. I just want to take one aside because you might be thinking that 61 genes is actually a pretty small number of genes to be induced by a major signaling event. But actually, when we do this experiment, I saw many more genes change over this time course. But interestingly, when I compared these to the mock washout conditions, we see that most of these genes are actually induced just by the change in the media themselves. And only really this top group of genes here, these 61 genes, are specifically induced with response to Notch. And there's a few other pieces of evidence that really suggest that I could focus just on this group of 61 genes. That group was the only group that was really enriched for having Notch binding proximal to their promoters, which suggests direct regulation by Notch of this group. And also, not only are these genes significantly induced in my TT-seq data, but also in RNA-seq that was collected by our collaborators as well. So now that I had this high confidence list of direct Notch targets, I could start asking some of the more mechanistic questions that I laid out earlier. So the first question was, does Notch activity affect chromatin accessibility like you might have assumed from previous work in the field? So to address that, I used an approach called ATAC-seq to identify regions of open chromatin in the genome and measure how accessible they are. So I did this approach at a few different time points after activating Notch signaling by GSI washout, and again, did all these mock washout controls to make sure that what I was seeing was specific. So the first thing I looked at is, now that I know where Notch is going to be active in these cells, these 61 genes, what do their promoters look like before Notch is ever activated? And what I saw is that in the reference condition, the promoters of these Notch upregulated genes are actually about as open as any other unchanged gene in the genome and much more accessible than inactive genes, which suggests that Notch is actually pretty limited to promoters that are already accessible in the genome, and is sort of, again, inconsistent with it acting really to promote accessibility. But to measure that directly, I then wanted to see what happens over time at the promoters of these Notch responsive genes. And what I saw by looking at the ATAC-seq data, here showing just a metagene, so looking at the average ATAC signal around the promoters of these genes, is that the accessibility of these promoters does not change at all over the time course that I measured up to 4 hours after inducing Notch signaling. And if we count the total number of reads over these regions, we see that there's no significant change overall. So this tells us that Notch is able to activate the expression of these genes without any increase in their promoter accessibility, and these promoters are already accessible before Notch is induced. But we know that Notch doesn't only act at promoters, it can also bind at enhancers. So I wanted to see what happens at the enhancers where Notch is active. To do that, I first had to identify where the enhancers were, and I don't have time to go through that now, but here I'm just showing H3K27 acetyl chip, which shows that these Notch responsive enhancers are in fact activated in response to Notch signaling, and we know there's direct Notch binding at these enhancers as well. But in contrast to this nice increase in K27 acetyl signal that we see over time, there's no significant change at all in accessibility at these enhancers either. So that really shows that Notch can activate both enhancers and promoters without increasing their accessibility at all. So that suggests that really Notch is not acting so much like this blue transcription factor in the cycle. But this was sort of confusing because I needed to then reconcile this with the previous work that suggested that Notch activity really requires switch SNF for remodeling activity. So to directly test that, I used the recently developed BRM14 allosteric switch SNF inhibitor to inhibit switch SNF activity and see how that affects Notch signaling. So with this inhibitor, I did the TTC kind of TAC-seq experiments again to see how it affects both gene expression as well as accessibility. So first, looking at the TT-seq data, this is the TT-seq that I already showed you, just in the absence of the inhibitor, where we see that these Notch genes are induced. And we see now that when we activate Notch but keep switch-SNF activity inhibited, that there are actually about 2/3 of the Notch target genes are dependent on switch-SNF activity for maximal induction. So this is consistent with the previous work, but it was a little bit confusing given what I've already shown you that we don't really need there to be any increase in accessibility for Notch to activate these genes. So I wanted to investigate what differentiates these two classes of genes to see if I could figure out what was the molecular basis for this dependence. So I first thought maybe the switch-NIF dependent genes are actually a little bit more closed before Notch is induced, and that was not true. These two groups of genes are just about as open as each other before Notch is induced. And we see for neither of these subsets of genes is there any increase in accessibility after inducing Notch. But what was different is actually how these genes responded to switch-NIF inhibition. So if you look at the promoters of these genes, the switch-NIF dependent genes, just when we inhibit switch-NIF itself, so I haven't even induced Notch in these conditions, we see that these promoters are actually sensitive to switch-NIF inhibition and the promoters themselves close up. Whereas the switch-NIF independent promoters are a bit more robust and are able to stay open even in the absence of switch-NIF activity. And this is significant if we count the total number of reads over these regions. So what this tells us is that it's not that Notch requires switch-NIF activity to promote accessibility in response to Notch signaling, but that switch-NIF activity is actually sort of required before and during Notch activity to promote accessibility at these promoters and also at enhancers, which I didn't have time to talk about, so that Notch is then able to act at these regulatory elements. So far I've done a lot of telling you things that Notch does not do in its role in promoting transcription. But the main question is still, how does Notch actually promote transcription then? So to answer this question, I use an approach called ProSeq, or precision run-on sequencing, that tells us where Pol II is across the genome. So ProSeq looks a little bit different from other gene expression assays. So I'll just quickly show you an example browser shot. So what we can see is that in ProSeq, you often see this peak of ProSeq signal right by the promoter, which tells us where paused Pol II is. And then for genes that are actively transcribed, you see ProSeq signal throughout the gene body as well. So I performed ProSeq over a variety of time points after inducing Notch signaling. And first, I wanted to see for these Notch-induced genes what their profile looks like before I induce Notch. And what I see is that at the promoter of the 61 genes that are going to be induced by Notch, we already see a large peak of paused Pol II at their promoters before Notch is induced, and there's very low signal within the gene body. And then over time, after I induce Notch signaling, we actually see a decrease in the promoter reads and an increase in the gene body reads, which is consistent with release of paused Pol II from the promoter into the gene body. And that continues over time, where you see a slight recovery of some of the promoter reads, but overall, mainly an increase in the gene body reads. And this is true also when we count the total reads over these regions and do some statistics. So you can see that if you look at the gene body reads, these genes are transcriptionally induced. You see an increase in the gene body ProSeq, but you do not see any increase in the promoter ProSeq. And in fact, you see a slight decrease, which is not really consistent with any models where Notch is promoting Pol II recruitment to the promoter, but is really more consistent with Notch promoting pause release. And to directly test that, we calculated what's known as the pausing index, which is the ratio of promoter to gene body reads. And you can see that there's significant decrease in the pausing index over all these time points, which is consistent with Notch really acting like this pause release factor that I showed in the cartoon earlier to promote these final stages of the transcription cycle. And this is sort of interesting because this is consistent with what a lot of other signaling transcription factors actually do in promoting pause release. So one more piece of data that I want to show before I wrap up. I had mentioned earlier that there were two classes of Notch target genes, ones that are switch-SNF dependent and some that are switch-SNF independent. And what we actually saw was that the switch-SNF independent genes actually have even higher pausing than the dependent ones. So this is actually consistent with the possibility that pause Pol II itself is actually what's promoting accessibility at these switch-SNF independent promoters and is helping to keep these promoters open in the absence of switch-SNF activity. So overall, this has led to our creation of this pause release model for Notch-dependent gene activation, where before Notch is activated, both the activity of the switch-NIF complex and pause Pol II itself maintain accessibility at Notch-responsive sites. And when Notch is activated, you get the full formation of the Notch transcription complex that leads to the release of pause Pol II into full elongation. So with that, I'd just like to thank everybody in the Blacklow and Edelman lab for all of their help on this project, our collaborators in the Astor lab, the Nascent Transcriptomics Core, and thanks for your attention. I'm happy to take any questions.
So thank you, Julia. That was fantastic. For people with questions, you can type them into the Q&A box, or you can click the raise hand button and I'll unmute you to ask your question yourself. I'll start off with the one which is, how generalizable do you think this mode of active, like rapid gene activation is in terms of the chromatin state? Like, is it kind of a general feature of rapid induction that you're not changing chromatin accessibility?
Yeah, so I mean, it's been shown for a lot of other like signaling transcription factors that they work similarly to this. Like this is how heat shock factor acts. So I do think for a lot of things that need to be really fast, this makes sense. What's interesting to me is that in some of the cases where Notch is active, you don't really need it to be super, super fast. So one of the things I'm really interested in is why is Notch acting in this way? And is this a common mechanism that it uses across a variety of cell types? And like what features does this mechanism actually give Notch signaling?
And then, so there's a question from Megan Isbin who says, do you have any evidence about whether Notch interact with PTEF-B or VIX, DSI/NELF, or how does it impact Pol II pausing directly?
Great question. That is something I'm planning to work on in my future lab. I don't have any evidence for that yet, but that's definitely a series of experiments that I have planned for the future. Thanks.
And then there's a question from Olga who says, hi, great talk. Considering that gene activation by Notch is dependent on already accessible regions, is it possible for you to broaden your findings to other contexts, such as cell type specific responses to Notch activation, including pathological contexts?
Yeah, so I think my findings sort of give a molecular basis. So it's already been suggested that this chromatin accessibility is really why Notch has such different gene targets and different cell types. And I think my findings really sort of highlight the molecular basis for that. So I think many people have been trying to identify or predict Notch target genes based on chromatin accessibility in different cell types. And I think what we've seen that Notch is not able to go to closed chromatin is really makes those predictive tools pretty powerful. But yeah, I think in the future, going into other systems where Notch signaling is either oncogenic or in developmental context, I think would be really interesting.
Great. Okay, so I think that's all the time we have for questions. There's another one in there which you can, Jelena, you can answer with the text in the text box afterwards. But thank you very much for the wonderful talk.